scientists whose work she respected. And none of those people could reproduce it either. So she really wasn't confident in the findings. She didn't believe that the findings were real. She couldn't reproduce them. She wasn't happy with them. So she sent out a letter saying that she wanted to retract the paper. But because the paper was in Nature and because of Nature's policy at the time, all of the authors had to sign that retraction paper in order for the paper to be retracted. But because the Czech authors said that no, they were confident in their data, they were happy that it worked, they had seen it reproduced in their own lab when she asked them, they didn't sign the letter, so the paper wasn't retracted. So instead, what Professor Fazey did was her own group sent a correction letter, a paper essentially refuting the findings of their 1994 paper. And they did this in 1996. She issued this kind of correction paper. She termed it a partial retraction. But of course, the original paper still remained as a full paper because the Czech authors wouldn't agree to retract it, so Nature wouldn't retract it. And she kind of continued her campaign against the paper. She really made it known that she wasn't happy with the results. She didn't believe in them. And she actually wrote to an ethics committee in the UK. And she said, this is just an extract from what she said. She said, it remained very desirable for Carl to determine why, during his time in my lab, he could repeatedly make preparations of recombinant rat NKRP1 and get the ligand binding data published, whereas my colleagues could not reproduce them either with the very tiny amounts of the protein that he sent us after his departure, nor with the recombinant protein de novo generated in my lab during the subsequent 4 years. So they had really spent 4 years attempting to reproduce this experiment. They had used exactly the same samples that Karol Bezuschka had been using, and they also had generated their own samples, and none of these showed up a positive result. None of them showed any of this binding that Karol Bezuschka was able to show was so consistent. And so this got the attention of the British Medical Research Council, and they launched an investigation. And during that investigation, they did find some irregularities in the data, and they noted that it hadn't been reproduced, even though it had been tried for up to 4 years now since the paper had been published, and nobody had been able to reproduce it. But ultimately, they weren't able to actually find any conclusive proof that anything unethical had happened or that there was definitely manipulation here. So ultimately, what they said was that it essentially comes down to being a scientific dispute, and it needs to be solved scientifically. They need to come together and do the experiment once and for all and decide whether it's reproducible or not. But the Czech group said that no, our data is fine, we can reproduce it every time here. And Professor Fazey by this point had also said that she and her colleagues had wasted enough time. They'd spent 4 years trying to reproduce it and they couldn't. So they essentially said, we've done everything we can do, we give up, we've issued our corrections, we've made it known that we're not happy with the results, we're going to move on. And in her response to the ethics committee, she said, yes, my group indeed made attempts to reproduce the results over 4 years. In the end, all we could do was to have a retraction paper in JBC, a partial retraction termed correction in Nature, and a retraction paper in BBRC, retracting the published observations on CD69. Carol and Miroslav declined at that time to retract the Nature paper as a whole, this is why by rule I could only correct or retract the parts that my group could evaluate in the absence of bioactive recombinant NKRP1. So essentially she disavowed herself of any of the results that she was allowed to, but ultimately the paper remained. And so at this point, some time had passed. Bezuschka had moved to the Czech Academy of Sciences, and he started working with this guy on the right, whose name is Vladimir Kren. He's not quite a main character in this story, but he's definitely a very important supporting character. And they worked together for a long time. They worked together for many, many years, and they both got along very, very well. There was no issues. In fact, Vladimir Kren was known for saying that he thought Bezuschka was a brilliant scientist. He produced amazing results, and he seemed to be very diligent. He kept good notes. Everything seemed to be great as far as he was concerned. He really had no qualms about working with Karol Bezuschka as a scientist. He believed he was a perfectly legitimate, above-board guy, and they produced quite a lot of papers together. And in 2012, Vladimir Kren went to a conference where he met this Professor Fazey. This was a conference in Lisbon. They met each other, they were talking, and they realized that both of them knew Karol Bezushka, and they began to talk about their work. And Professor Fazey then brought up the fact that she had had this disagreement, this dispute over the 1994 paper, and that she didn't believe the results. She thought there was something fishy going on. So they begin to discuss this, and they begin to talk over it in detail. And Vladimir Kren is convinced that, yeah, something's not right, and he agrees to essentially set up another investigation. He says that he will go back to his lab in Prague, and he will talk to Karol Bezushka. He will say, give me your lab notebooks, tell me what you did, and we'll reproduce the experiment, no problem. So yeah, he went and he talked to Professor Bezushka. He got the original notebooks from back at the experiment. Professor Bezushka still had all of these. He was, as everyone believed, was perfectly above board. They took it. They agreed that they were going to document their investigation comprehensively. They would, at every step, they would take detailed notes. They would take photographs, videos, anything that was necessary to finally get to the bottom of what was going on. But they also agreed that they were going to do it independently of Bezushka. He would have no involvement in actually carrying out the experiment, but that he would be willing to cooperate. If they had questions or if something wasn't clear in his notebooks, then he would happily go along and help them, and he would give them any guidance or any clarifications that they might have needed. And so they begin doing the investigation. It's mainly being carried out by 3 students of Vladimir Kren. And initially they get some mixed results. They get a lot of negative results. They don't find anything for the moment, and they think, okay, it's, maybe we're having some technical difficulties. We keep trying for a while. And then suddenly they start to see a couple of positive results. So they start to see, actually, maybe this experiment is working. Maybe there is something here. And they do begin to see the binding. But one morning, something kind of sets off an alarm bell in one of the students' minds when they come in and they find that the fridge where they keep the samples overnight wasn't the same as where they had left it. So there were signs that things had been moved or that they'd been opened and tampered with. And so they got quite suspicious quite fast, because they were trying to be conducting an independent investigation here, and suddenly now it seems like someone is interfering with that investigation, which very quickly made them suspicious. So what they did is they actually came up with a very kind of clever plot or trick, whatever way you want to put it, where what they did was they prepared test batches as usual. They made however many runs of however many samples they needed, and then they took half of them and replaced them with just plain water. So instead of having a solution with the proteins and the NK cells that were supposed to be binding together, half of the samples were just water. So they took half of the real samples, put them in the fridge, the other half were water, and they put them in the fridge, and then they took the remaining samples, the real samples, and they put them in another fridge in another lab, and they told nobody. So they left these overnight, and they waited to see what would happen, and they came in, and every single sample in their fridge was positive. It was showing up that the binding was happening, that they were getting a positive result. But the problem was that half of those samples were actually just water. So there was no way that their test should have been showing up a positive result. And the test that they were doing, by the way, was they were doing something called a radioligand assay. So they were binding assay. So they were basically putting a radioactive marker into the proteins that were supposed to be binding. And then when you separate out the proteins at the end, you test how radioactive the resulting product is. And then if you've got radioactivity, then you know that something has bound to your protein, because the protein itself isn't radioactive, but the binding agent is radioactive. So if there's radiation that wasn't there before, well, it must be coming from the binding. But now when they were testing, even the water was coming up as being radioactive, which of course, again, confirmed the suspicions that something was being tampered with, and that something really not right was going on. So to fix this, they decided that now that they were absolutely sure that someone was messing with their results, they set up a hidden camera. They set up an infrared camera, a night vision camera, And they left it rolling. They set up their samples, all real samples this time, put them in the fridge, left them overnight. Except they didn't actually go home. They just stayed and watched the camera on a live feed, a closed circuit camera. And lo and behold, in the middle of the night, they found Professor Bezushka had come in, had broken into their lab, which was, essentially trespassing on their lab. And he had opened the fridge and he began to take out and tamper with the samples. So it was Professor Bezushka himself was now caught on camera, fiddling, messing with his own the investigation into his own work. And at this point, what they did was because they were still in the university, they ran back down to their lab. But by the time they got there, all they saw was, as they claimed, they alleged all they saw was a figure in a white lab coat running down around the corner at the end of the corridor. So they chased after this person. They were 100% sure it was Professor Bezushka because they had seen him on the camera in their lab just a few minutes ago. They knew he was tampering with the results to try and fraudulently misrepresent the results that were coming. And as they were going, they couldn't find him, but what they found was a sleeping bag in one of the rooms on their hallway, on their corridor. So they found that Professor Bezushka had apparently been sleeping in the university, unbeknownst to anybody else, so that during the middle of the night he could come and break into this lab, open the fridge, and mess with the samples, mess with the results. So eventually they continued walking. At this point, they called the director of the institute, director of the Czech Academy of Sciences, and they said, we've got someone breaking into our lab. You need to come very quickly. And he did. And they opened all the other buildings in the university and they did a thorough search. They called the security and they found Professor Bezushka in a nearby building just a few minutes walk away. And so they confronted him and they said, "What are you doing here? Did you break into our lab?" And he said, "No, I wasn't anywhere near that building. I've been here the whole time." So at this point, they hadn't yet shown him the video evidence, but then they brought out the video evidence. They found it and they showed him pretty clear images of himself in their lab, and he just again continued to deny. He said, "No, that's not me. That couldn't be me. Must be someone else." And at this point, they said they were getting quite annoyed at him. They said, "You must have been in the building." And he said, "Okay, yeah, I was in the building, but I was on a different floor. That's still not me in your lab. I wasn't in your lab. I was in the building, but I was looking for some other equipment somewhere else." And so he maintains his denial all night long. And then finally, the following morning, he admits, "Yeah, okay, I was in your lab, but all I was doing was just checking that you were doing the experiment properly." He maintained his denial that he was interfering with or tampering with any of the samples in the fridge. So at this point, there was an ethics committee set up in the university by it's a joint ethics committee set up by Charles University and the Czech Academy of Sciences. And at this point, they had already had some pretty slam dunk, incontrovertible evidence that he was fraudulently manipulating results. But Bezushka, again, still maintained his innocence. And actually what he did was he set out and he started to launch a really pretty vicious attack on Vladimir Kren. He accused him of being abusive towards his students and forcing them to find negative results. He also accused Kren of having collaborated with the secret police when the Czech Republic was under a communist rule. And he actually even accused Kren of selling out his own father to the secret police. And he was generally trying to make the case that Kren was totally untrustworthy and this was all just a personal attack on Bezushka, and Bezushka had done nothing wrong. But the ethics committee just didn't believe anything. There were completely damning findings against him. And he was disgraced, he was thrown out. And the ethics committee, in their findings, they sent this letter to Nature, to the journal. They said the ethics commission, on the basis of their findings, declares that Professor Karol Bezushka, with the highest probability, committed scientific misconduct or dangerous or irresponsible deviations from accepted practice in how to perform research. And this scientific misconduct occurred repeatedly. So they completely just threw him out. They said all of his work was completely untrustworthy now. It was essentially to be taken as fraudulent. So Bezushka was fired. The 1994 paper was finally retracted in 2013, 19 years after it had been published. He lost his positions in Charles University. He had actually been promoted as high as the head of the Faculty of Science in Charles University by that point. And he also lost his position in the Czech Academy of Sciences. So he was, he was completely took a massive fall from grace. Everything he had lost. He never worked in academia again. And then he was actually subsequently sued for defamation by Vladimir Kren over the accusations that he had made to the ethics committee, all of which were subsequently found to be completely false. The students all testified that they had never been abused by Vladimir Kren. Kren's own father said, no, I was never had any interaction with the secret police service in the Czech Republic, so I don't know where that story came from. And the court ordered Bezushka to give Kren a full formal apology. He was also ordered to pay legal costs and legal bills and all this. So he, everything that he had done was completely thrown back against him. He had lost everything. And then, they begin to look back at all of his other works, and suddenly people cast a much more kind of sharper eye over the results that Bezushka was involved in producing. And suddenly lots more papers began to be retracted. And at the last count, I think it was 11 of his papers had been fully retracted and were completely struck out of the record because they were found to be not just incorrect, but completely fraudulent and manipulated results, essentially completely made up by Professor Bezushka. So, is that really the end of it? Is it all just a nice happy ending? I mean, the record after 20 years, even if it took 20 years, the record got corrected. Everyone was, everything has been set back to the right state of affairs, so to say. Yes, it took a long time, but eventually the right answer was arrived at. But then equally, you could ask, did it really need to take as long as it did? Because Professor Fezzi and her group had noticed issues, even as soon as 2 years after they initially published it. They realized that they couldn't reproduce anything. They went straight to the journal. They said, we don't believe the results. But the journal said, well, unless everyone agrees, all the authors agree, then you can't retract the paper. And equally, because it lasted so long, then it's possible that there was a huge knock-on impact. There was a lot of wasted time. One of Professor Fezzi's big complaints was that she had wasted 4 or 5 years of her and her colleagues' time trying to reproduce fraudulent results. And there was, this was a heavily cited paper. By the time it was retracted, it had something like 275 citations, and most of these studies were building on what turned out to be completely falsified data. And because he had gotten away with this falsifying his data, and he had built a career on it, he'd gotten himself to pretty high positions in his university in Prague. You saw that he had at least 11 papers retracted. So he was clearly, once he got away with this paper, he was emboldened maybe to do more of it. And it seems like his work was being allowed to remain part of the scientific record for so long that people were essentially just wasting their time trying to reproduce or trying to build on research that was completely false and that just couldn't stand up to any sort of scrutiny. So yeah, that's the end. Thank you.
Great. Thank you. So you can unshare your screen. John, that was terrific. Thank you. Very much resembles the Sean scandal story. It didn't take 20 years. It was much faster, the fall. But the same kind of motivation. You always ask yourself, what would bring a scientist to do this? It's just unbelievable. I just wanted to comment first on the topic itself, the first paper you introduced, where the issues came. Nowadays, we have CAR T therapy. That's exactly the promise. You can re-harness the immune system to attack cancer cells. That is very difficult. Nowadays, what we do, we take cells out of the body, we re-engineer them and introduce them again into the body, and then they can attack the tumor, because usually the tumors develop defense against the immune system and basically they evade the immune system. So it's very hard. And they came up with a story that you just need small sugar and you can do it. Well, it's much, much more complicated. But of course, that paper, I guess, generated a lot of noise in the system because the promise was fantastic. We nowadays know that it's much, much harder to do this. I want to open this for discussion. Any thoughts? Just turn on your mic and participate in the discussion. Go ahead. What is similar? What is different from the Sean case? Don't be shy. Turn on your mics.
There seems to be more efforts to cover the act in this case.
I mean, tampering with the validation experiment later on, that's going beyond Sean.
Right.
This is absolutely criminal. So in terms of personality and how far you're willing to go. More? I really hope that what we are doing here during this semester is some food for thought. And what we are trying to do is to make sure you guys know and aware of things like that. And when you do your science, nobody will come with any complaints. It's, to me, to these days, each case where people do fraudulent science is incomprehensible. I just don't understand the motive. Yeah, people want to have an edge. People want to, be promoted and get recognized and so on. But are you willing to go so unethical and maybe criminal to do this? We are not trained as scientists to think this way. It's just too outrageous. But every now and then, and I introduced a case, just was on the cover of Nature like a few weeks ago. Just cannot comprehend this.
It's no different to the need for us to publish or for any of us to look at our futures. It's the same need or the same desire. The goals are the same. People have their own moral compass. Then if you remove that from it, so theoretically it's very understandable. The same way you'd go and take your neighbor's car if you could, or you didn't feel the law wasn't there to stop you. So some people need the law.
But we want to think that science is a little bit better than the rest. Yes, in any aspect of life, people take advantage of other people by cheating, by committing crimes, by doing all kinds of things. And yes, it's at the end of the day, it goes back to your own upbringing and possibly genetic makeup. This is a very debatable issue, but let's say upbringing. And
so why would it be different?
Yeah, that's exactly the point.
Bankers, politicians, or anyone else.
That's exactly the point. And we have to recognize it. The scientists are as everybody else, although we are trying to think that we are better. So we are not. Any other comments?
I think it's acceptable.
Leon, we cannot hear you.
Sorry. Better?
Yeah.
I think it's okay to have some doubts about the scientific idea, but maybe it's running a bit too fast to run in assumption. But the same as the Sean scandal, this sounds like mental problem of the scientist. And how can you spot?
I can share with you guys that I had in my lab many years ago somebody with a bipolar syndrome. It was hell. It was very difficult to handle. And there were all kinds of things done, but not scientific fraudulence or misconduct. That wasn't his thing. But yeah, you never know. And again, as a PI, you have to trust. As a collaborator, you have to trust. And you cannot go up front and introduce some doubts in a very blunt fashion. We are basing our work on trust. And this is why it's so difficult. when you have a paper where several co-authors write a paper and every person was in charge of one part, and you're not expert on that part, how do you know to trust that somebody is not kind of trying to push something that is wrong? So you need to be careful, and then you need to do your utmost to understand what the other guy is doing, to ask difficult questions. But it's very hard. Once trust is being violated, all hell goes loose. And I've been doing science for over 30 years, and till this day, I'm afraid that, someday one student or a postdoc or whatever will do something wrong, and the responsibility is mine, not his. So how to make sure that this doesn't happen to you? And yes, in the population of scientists, you always have the outliers that are as in the general population. There are the cheaters, the criminals, the mentally sick. I mean, could be many different things, but you have to be very careful. Yeah, Sean lost everything, and this guy lost everything. You're driven to do things that are non-kosher. Well, I tend to think that, you cannot lie to all people all the time, and eventually you'll get caught. But it took 20 years. So, okay, I think we discussed this case enough, and now we are switching to David. So, David, you can share your screen. Where is David?
Here I am. Oh. 1 second. I do hope that will work. Okay. Sorry. My computer is...
Okay. Yeah, we can see.
Yes. Yes. 1 second. I'll just return to the first slide. Okay. Can you see the...
Yes.
The top? Oh, okay. So, today I will try to talk about batch effect in science. It's not scientific misconduct as it arises in problem in science. And if I may, I kindly suggest to put the Zoom screen in the upper left part of the screen. I'll try that most of the slides would be left this part clear. So to start, I will talk about a paper from 2014, a comparison of transcriptional landscape between human and mouse tissues. Too long, don't read. What we know, mice are commonly used in science and used as a model organism to study.
David, just one second, David. Your audio is not that great. I don't know, it's because maybe you're moving your head or whatever. Try to speak to the mic in a steady way.
1 second, maybe. Check.
It's better.
Now it's maybe better?
Yeah, much better.
Okay, so, okay, I'll resume. Okay, so mice are commonly used in science, and almost all genes in humans have a mice homologue that creating available assay to study mice and hopefully find new cures and treatments. This article claims that human and mice are not so similar as we thought and actually quite different. So let's start. Mice is a very common model organism in terms of anatomy and physiology. Most of the organs and they function similar to humans. Most of the genes, more than 90% of the genes in humans have a mice homologue, and the mice used in most research labs or in research labs and drug development, and many Nobel Prizes were won using mice, and as I said, they studied across the world. And another thing is mice is very relatively easy to grow and to handle and to change the genome. The second part that I need to introduce is RNA-seq analysis. RNA-seq analysis, first, the central dogma that I assume that all is familiar with is the DNA, the genomic information contained in the nucleus as DNA molecules, and the DNA is transcribed to RNA, which go outside the nucleus to the cytoplasm, and then translated to proteins. RNA-seq analysis is attempts to catch the RNA, the DNA molecule. It can be said the DNA molecules that the cell wanted to express and turn to proteins, and study them. So we can learn about the current cell state. Usually, it's not so hard procedure. We take in a tissue, we extract in RNA segments, we are fragmenting them, adding and using a sequencing machine, usually from Illumina family, and we get transcriptome files which we can analyze. Okay, that's all. So back to the article. In 2014, Xin Lin, the main, the first writer from Michael Sinder group, they both from a mouse ENCODE consortium from Stanford, found the next finding. When they perform RNA-seq analysis on multiple, actually large cohort of samples from human and mice from different tissues, they don't see any similarity at all between the tissues. What I mean, usually we could think that the brain tissue, for example, in human, would be similar to brain tissue at mice because they have similar genes. We did know that we thought that they express similar levels of proteins, but their claim that they are just clustered differently. All tissues, here as cones, are clustered different as mice. Mice clustered differently than the human ones. This was in all cohorts, and only cohorts that were from Stanford. That was their main claim. Okay, so in theory, they use standard procedures for RNA-seq preparation. Possibly they used too few replicates in their data, but they checked every tissue, and still it clustered differently between mice and humans. The article is still cited today, 2022. I checked it, and they're actually not alone in this debate. Yanai et al. Presented here found a similar thing. He found that human tissues and the mouse tissues are clustered differently. Actually, it's not cited at Lin Work, which raises questions why it happened. but that's a different story. And Seok et al., which found very similar thing. I will present his article a bit later in the presentation. So just to remind, this is a protocol of RNA sequencing, standard protocol without a change. So in 2015, Yoav Gilad, a geneticists at Chicago University, published the following tweet. I hope that everybody can read it. They reanalyzed the data from the Lean article, and they found the following. It's a bit hard to read. Okay. So what they found is a huge batch effect. Here, see if we can. Okay, no. Accidentally I removed this photo. They found that their data that Lin et al. Used was actually from five different sequencing machines, and as you can see, some were from just mice in red. No, sorry, this is human. So some just human, some is just mice, and some were both mice and human. To explain what batch effect is non-biological factors that cause change to the data. What I mean, for example, different people performed the experiment, or the data was performed on different days, or different kits were used to data process, or different equipment, or different location in the world, which actually was in the case of a Lin et al. Article, or in our lab it was a different PhD, what the PhD student ate for lunch. So it actually happens. So after the Gilad founding, there was many different responses from Twitter, which gone from how could they not check for batch effect? Everyone need to check them. Why the PNS didn't check it, or the PNS reviewer didn't check? No bioinformatician reviewing for PNS, and so it goes on. And I must state that Gilad first published in Twitter, tweeted the result, and then after a few months, after a week, he published it in open in a journal that you can review after the publishing. I'm not sure. Yes, you can peer review after the publishing. So there was two similar articles similar to Lin et al. One of them is greatly example here. The same data, 1 year apart. Seok et al., which Seok et al., which was cited by Lin, and the same data that they claimed very different results. Okay. So batch effect is a very serious problem, and it influenced even the 1000 Genomes project, an article from 2018. And I'm a bit short of time. Okay, so what was actually the batch effect that Gilad found? For that, a bit quick introduction. Each RNA-seq file, each transcriptome, each RNA-seq file is saved as FASTQ format file, which consists of header, the sequence himself of the genome, another hazard, and a quality score. What important is the hazard. What they found is it was the machine ID number. And they noticed that that each sample, if they check, they had 5 different machines that created the data. And when they account it, if they use they try to reproduce the data. If they didn't account that the data was from 5 different machines, they get a very similar result to Linet Al. Here you can see that, okay, it may be too little, but in red, human, in blue, mice, that human and mice are quite different between one and another. And even here, human and mice are different one from another. But when they use removed the batch effect, they see that the tissues cluster very similarly one to another, and similarly here. So the data probably contained batch effect. So should the data was retracted? There are few people that I'll give their responses to it. Leo Petcher, bioinformatician, that said, yes, definitely, the article should be retracted because it contained the batch effects. But different bioinformaticians said, actually, it should be retracted, but all papers have some batch effect, or the design is not optimal, so it should be left to question. But the main point is that Twitter is appropriate platform to raise this question? I remind that Gilad first published it in Twitter, not in any journal. The Gilad answer actually to this was, it was the only way. People that challenge results from ENCODE or similar large consortium are never published in the same glamour journals. He didn't have any option. But on the other side, Cinder, which was a PI, there are a proper way. I don't think that engaging in this medium would help us. And there was an avalanche of comments before we even knew about it. And the Cinder et al. Group claims that the batch effect did not alter the findings. They replicated it in another set of data and didn't see the effect of a batch effect. I do want to emphasize a few sentences that Yoav Gilad replied to Shin Lin and Cinder. It was the article. It was extraordinary because it was counterintuitive, because it challenged a strong paradigm in biomedical research. I want to remind that mice is a model organism. The claim was that mice shouldn't be a model organism. So it challenged a very intuitive paradigm. And you may have assumed that this type of batch effect is minor, but many others have shown that it's actually quite considerable, and it goes on. So, in summary, mice used around the world as a model organism. In 2014, Shin Lin published data that claims that mice and humans differ. In 2015, Yoav Gilad tweeted that the data contained batch effects. The batch effect resulted, probably, from using different sequencing centers and different protocols for human and mice samples. After removing the batch effect, the main claim of Lin article disappeared. Lin et al., while collaborated initially, they, as I must say, I didn't say, they were who provided Gilad with their data. Still claims that the batch effect do not influence the data. The paper is not retracted and still is cited. I checked, I think about 12 or 13 less papers, and everyone is still citing the paper and still looking at it as the debate is never up. And the debate status is unknown. So that actually everything. And I would like to open a few questions for discussion, if I may. Is it actually Twitter is a good debate platform? As on the one hand, it actually Gilad had no other option to in his hands, but on the other hand, it's social media. Does the paper should be retracted? Currently it misleading scientists who is actually right in this debate. There are people that did work, did try, and it happens that some articles, some findings are not similar to one another. How can a batch effect can be prevented? And if it should be addressed that the paper is still cited. So that's all.
Okay. Thank you very much, David. This is terrific because you can answer your screen.
Yes,
We heard quite a bit of stories about fraudulent science. This is not. Here we have a technical question, how an experiment is done. Not all of you are bio-oriented. Some of you are physicists or engineers. So we don't need to go and dive into the actual technical issues. But basically, Gilad says that they did the experiment wrong and they interpret the result wrong. That's the most important part. And there is an argument. And I think the point about Twitter is extremely important. I brought it up during my lectures. I've shown you that Arnold Francis immediately went to Twitter when she found out that her paper was fraudulent, and she retracted it. But there is an official way of doing this. Gilad could have written a comment to the editor, which would be published, and the editor would go back to Snyder and Lin and ask them to respond to that comment. So usually you can find in all respectable journals, at the end of each issue, there is comments and response to comments. So that was the old way of doing this. Nowadays, people don't wait. You immediately go on Twitter and the whole discussion starts. And I don't think there is a way to prevent this. So I call this social peer review. And as we discussed many times during the course, the whole concept of peer review, first of all, doesn't work, really. And second, it's in flux, it's changing. And what would be the role of social peer review in the future? You can still do all the official things, you can write a comment to the editor and so on, but people will immediately start discussion. Papers go out and immediately there is a discussion, and people comment. And so I don't think we can stop this. And I think a better question is, can we use it to do better science? So we'll see, time will tell us, but thank you, David, for bringing up a different type of issue with, proper conduct of scientific research. The issue here is really, did they do the experiment right or wrong? Did they have the right controls? They could have avoided the batch effect and much more clear and clean and so on. So doing good experiments with good control experiments is essential. And apparently that was not done here. Open for discussion. Turn on your mics and tell us what you think about this.
So I have a comment about this. I mean, this is just incorrect use of the technology.
Yeah.
It's pretty much like basing, calculations on the fact that 1 + 1 = 3. Like it's not valid in any way.
They are doing principal component analysis on data that was not taken properly, all on the same machine, same day, same this, same that, right? So the principal component analysis shows the batch variations. That's what they do. So yeah, and they claim that they have redone this, and I think Gilad, if I understand correctly, Gilad showed that the analysis really showed the batches. And so it's hard for somebody to acknowledge that something is wrong, but I think it's much better policy for your own career, if there is a doubt, to be much more cautious and accept that something was done wrong, and rather than insist that everything you've done is correct, because if later on the jury is still out, but this, and as David said, I mean, we are not sure what the status of this argument, discussion and argument is still today. But in the last case that John was discussing, it took 20 years. So maybe in 20 years, we'll have an answer for this debate as well. More comments?
I think that speaking about social media and the way we take the knowledge now, 20 years ago, there were papers and no comments.
There were comments. There were comments. They were not as common. But if you write a paper from somebody and you have very strong objection and you thought that something is wrong, you would send a comment and the editor should have published it. And then the editor would go to the original authors and say, you have to respond to this.
Right. It's more complicated process than putting a post in
Of
Facebook
course.
or Twitter. It's different for the good and for the bad, right? It has two ways to look at it. When I look for a paper or for knowledge, I only have the impact factor of the journal, what I think about the journal, and maybe how many times this paper have been cited. And I think now I get it that it's not enough, because for me, reading a paper like David presented, I don't have the tools and the knowledge to criticize it. So maybe one day we would be able to see those Twitter debates under those papers, or to feel a bit more about.
So some journals started to compete with Twitter. They open a page of discussion after the paper was peer-reviewed, published, and so on, and people can go in and basically you see an ongoing discussion about this paper forever. Again, I'm emphasizing that this whole peer review concept is kind of changing in flux. Social media is going to strongly affect it. And we had many, many examples during the course of high impact factor science nature papers, which were fraudulent and retracted, or which were wrong, without ill intent, but wrong and retracted. And the old way of finding the mistakes was very slow. Maybe, we don't know. Jury is out, but maybe social media will help us find mistake faster. More comments?
I think it can also generate mistake faster.
Of course.
I mean, the coronavirus was the proof of that. People misinterpreted legit papers and turned them into false data that just scared the public. I think you can also
amplified wrong data.
Yeah.
The minute everything is out in the open and social media discuss things, you can very easily take a scientific result out of context and scare the whole public. So that's a whole discussion by itself. Yes, it happened during the pandemic. So as Omer said, social media has good aspects and bad aspects. And it's very hard to separate. Every new technology that was introduced to humankind over the past few hundred years had good things and bad things. And you can always abuse a new technology. So it's very hard just to pick up only the good things and prevent the bad things. right now, big governments, the European Union and everybody else is looking into those mega companies like Facebook and Twitter and all these guys and starting to put some regulations. In the mid-'80s, it happened to the phone company. For 100 years, Bell Laboratory, Bell, the phone company, was a monopoly since Graham Bell. And when they started to abuse the system, the Supreme Court in the US decided to break up that company to 7 small companies. We have many examples in the past. And I think all the big, like Google, Amazon, Facebook, Twitter, they are under magnifying glass right now. And I think things will change over time. They will have less power than they have right now because they are abusing it. How will that affect science? I have no idea, but we live that transition right now. More questions, comments? If not, let's move to the last presentation of today. That's Radhika, so you can share your screen.
Now you can see my screen?
Yes.
Okay, so hi, my topic, the paper which I'm going to talk about is visfatine, a protein secreted by the visceral fat that mimics the effect of insulin. So basically, the Shimomura, he's a Japanese scientist who claims that he discovered a novel protein which actually have the similar effect as of insulin. And why there is like a need of alternatives when we have the insulin. And so this article was controversial because it was like a new like a kind of like a second protein which is exactly works like insulin. And this article was published in 2005, and after like 2 years of publication, it was retracted. And it was published in the very famous Journal of Science, which has like a very high impact factor. So why this article was controversial, we will go one by one, but first let's go through statistics about the diabetes. So according to the WHO, in world, like right now, it's the 422 million people are affected or diagnosed with diabetes. And every year there is like 1.5 million deaths per year, which is like somehow related to the diabetes. And until 2045, they're going to be around 700 million diabetic, like people which are diabetic, and mostly they're going to belong to the population of adults. So these are like very, diabetes is like one of the most, like occurring diseases nowadays because of the lifestyles. So talking about the history and the cure of the diabetes, the insulin was discovered by Sir Frederick and Charles in the University of Toronto in 1921, and it was purified from the cattles of the pancreas. And from but it was like very old disease. It like started from 250 BC. It was like recognized, and like each passing years and years, like people try to cure this disease by insulin. And now there are like so many different types of kind of insulins, way of taking insulins in the body are available in the market, like insulin pens and insulin external pump and everything. But now, like in 2022, where we stand on the cure of the diabetes. So talking about that, after doing some research, I found out that basically there is no certain like exactly 100% cure for diabetes. Generally, people claim that either you can have a healthy lifestyle, do exercise, but there is no such like drug in the market who actually can properly cure diabetes. So giving a brief introduction about how these insulin work in the body. We acquire food, and that breaks down into the glucose, which goes directly into the bloodstreams, and where the pancreas, the beta cells in the pancreas produces the insulin, and the insulin activates some cells, fat cells and muscle cells, which take up the glucose, and that's how the glucose level in the body is maintained. There are two kinds of diabetes. It's diabetes type 1 and type 2. If you're talking about the diabetes type 1, it is basically an autoimmune disease in which it is generally often occur in the childhood, and it in this immune system attacks their own beta cells, present in the pancreas and stop producing the insulin. Talking about the diabetes 2, which is like more common, and it's 90% of the population have diabetes 2, the type 2 diabetes. So it occurs generally, it has like many factors, including the cells become resistant to the insulin, and the insulin stop working on the cells, and the other dietary problems. So this is like a basic mechanism to understand like how insulin works. And the insulin basically, insulin in the bloodstream binds to the insulin receptor, which is present in the cells. And after binding to the insulin receptors, the cascade of signaling start, and it activates the system. And the GLUT4, which is like a transporter of the insulin, it's translocate from the cytoplasm to the membrane, and the glucose, then the glucose starts to come into the cells through this GLUT4 transporter. So this is like a basic mechanism. So this group of scientists, what they said is, like they find exactly visfatin. They named this protein visfatin because these are produced by the visceral fat. So you can see this is like the visceral fat is just below the abdominal muscles, and the subcutaneous fat is a belly fat. So they claim that this new protein, they are produced by the visceral fat, like obese people. Like the more the obese people are, they will produce more visfatin, and which have the similar effect as of insulin. So this is what they claim. And they present some results regarding this new protein. They said that we take samples from 2 females from both the fat, subcutaneous fat and the visceral fats, and then they analyze it by sequencing and mRNA expression level, and and they also do the Western blots to analyze the protein expression. So they find that, as you can see, the mRNA level expression was very high in the visceral fat, and the protein expression was exactly same as the PBF, which is like correspond to a protein that is PBEF, that is pre-B cell colony enhancing factor. So they claim that this is like a new door to new protein, which can actually work like an insulin, and which is already produced by the visceral fat present in the human body. So they wanted to, there are some results which they propose that there is a link between the adiposity and this new protein. So they show that in the visceral fat area, they will have more of protein of this is found, and in the subcutaneous, there is no such protein. So it means that the more obese the guy is, he will have higher chance of having this new protein in the body, which can actually work like insulin. So for this, they also did some experiments in the mice, and they take 2 kinds of mice, one normal mice and one with the diabetic mice. And they showed that the diabetic mice has the more expression of this newly introduced protein. And also they have checked the expression level and everything. And then they found that in all of the visceral protein, they found the protein amount, the mRNA amount was higher in the obese mouse. So they're claiming the visceral fat produced this new kind of protein. So the more the fat person have the visceral fat, the more of the expression of this thing was present. And now to prove their point, they take a mouse animal. Notice that they only publish the data of the male mouse. They said they genetically altered a mouse, and they produce a heterozygote mouse, which they feed on. This one is the normal mouse, and they feed them on different kind of diets, normal diet and the high fat diet. And the high fat diet produce more visceral fat, and it gives more expression of this newly introduced protein. And they also have the more mRNA expression level. And while the normal diet, they don't have any this kind of protein is produced. So they wanted to compare how is the insulin, does it working as an insulin by lowering the glucose effect. So they actually injected these mice with the different levels of new protein, 15 picomole and 100 mole. And they said it is a dose dependent. Like the more you inject this protein, the more the glucose level comes down in the body within the 30 minutes. And they said this new protein doesn't have any effect on the insulin. So it's working independent from the insulin. And it's like also lowering the glucose in the body. And they also injected this protein in the obese mice, and they found that it has a similar effect as of insulin. So until now, they proved that this protein is produced from the visceral fat and from the obese mouse, and it works exactly as the similar insulin. And they also did some glucose tolerance level and fasting and feeding tests. And from that, they found that this protein has the same physiological effect in lowering the plasma glucose level. So this is also, they take different kinds of fat cells and they use the different kind of, different level of, different molar concentration of these two new visfatin and the insulin, and they inject and they try to grow these cells into this. And they also showed that this protein also activate the receptor of the insulin receptor of the cells, and they increase the uptake of the glucose in the cells. They also shows that this protein also activate the, like I showed previously, that it activate the insulin channel just like the insulin. And they also calculated its binding affinity, like how strongly this protein bind to the same insulin receptor. And they said it is 4.4 nanomolar, which is like similar to insulin, which is 6 nanomolar. So it, so basically in short, they said that this protein which they found is have exactly the comparable ability to activate the insulin signaling, and they can help the glucose uptake by binding to the receptors and also to inhibit the glucose release. They said that this new protein has a same effect as the insulin, and it lowers down the plasma glucose concentration in the bloodstream. And the visfatin may be useful for the target for the development of drug therapies. So this article was very promising, and it was one of the highly cited articles, but it was retracted after 2 years when it was published. Basically, their own committee of the Osaka University, their own committee asked them to retract this paper. So, but they still stand on their conclusive studies. They said that we produce this protein, and not all the batches are will work as we showed in the data. And the university was claiming that they are like hiding some data.
Right. Radhika, I'm sorry for bursting in, but I did not get it. What is the reason? Who raised the first criticism, and why the university had this committee, and why eventually they retract? So somebody was not happy. You did not tell us about this.
Yeah. So their university is Osaka University Graduate School has asked them to retract this paper, because there is like a 1 year because this scientist was involved in the one of the paper which was already retracted from the Nature Medicine.
So it was personality issue. Somebody on that paper was already suspected for other.
The lead scientist, like the group leader, was already have like a previous experience of retracting the paper, and it was from very good journal, that is Nature Medicine. And also the Nature Medicine claimed that this article, the problem was the reproducibility of the data. And the same thing happened with this article. Their graduate school only, the Osaka graduate school only asked them, this group, to retract this paper, and the committee was set up, and they have a committee going on. But this scientist was very aggressive about his research, and he said he wants to take legal action against the committee. So it somewhat looks like a rivalry also, like between, there was like no, but the scientist was, there was something fishy, because the scientist was already involved in the retraction of the paper, which was already like in the Nature Medicine, which was retracted. So there was already a problem with the reproducibility. And in this paper, they conclude that not all the preparation of this protein which they produce, they said they have some stocks of this proteins which they produce, and they are like ready to provide for the validation. And they also claim that not all the preparation of the visfatin people will produce will not have the similar effect. So it was not a very stable or good results to say, because if you produce a protein, it should be stable. It should work, or like it, maybe the condition will be different in something, but producing only 4 lots and saying that like not all the lots will work, it's like sounds fishy to also to the people of the committee. So this happens with this thing. That's why the committee, the Osaka school, the graduate school, raised the question to retract the paper. And also the Maesa Toyama, who is like a dean of the university, they said she doesn't want to public, like they have a committee for 1 year, but they don't want to make the report public. And but the reason was that they said that they hide the data. Actually, all the experiments were done on the male mice only, and they have the results of female mice, but they didn't publish it. So there was argument growing with the editor-in-chief and everything. So even the dean said there is no indication of the fabrication of data, but actually they hide the data, which was problematic because it will not give you a conclusive results. So Harvey Lodish, who is a biologist at the Whitehead Institute of Biomedical Research and the Massachusetts MIT, he said that he doesn't like have any problem like if he understand if he hide the female heterozygotes data, but still not a lot of people not agree with it. So in editor-in-chief, that is Donald Kennedy, we said that there is an investigation going, but they were reliable on the they were relied on the university final determination, but that guy was very reluctant that he will not take the paper back. So finally, after 2 years of this committee and this thing, he finally retracted the paper. And like I said, this was the paper he was involved in Nature Medicine. And it clearly mentioned that the results were not reproducible, and it is artificially presented in the paper. It means the results were fabricated. So under these circumstances, they asked the authors to, and he was also the lead researcher in that paper also. So the two top journals, Science and Nature Medicine, he published the paper, and it got retracted because of the problem with the reproducibility. And if you'll see the statistics of this paper, that it was published in 2005, retraction was done after 2 years. And you will see that how many citation it has, like the citing articles, like before retraction, the article citation was 232. And after retraction, it's like 1,200 almost. So the total citation was around 1,500. So it was like a very controversial article because of the high citation, because people are still using this article to prove their studies and points. So I seen the problem with this article was the reproducibility, because he said that he produced certain protein and he has very little amount of it, and not all the cisplatin batches will work. So it sounds very fishy to someone you can produce the protein, but only some amount of protein will work and some will not. So it was not a very stable or good results. Second thing, it sounds very that it's he gets into the competition to get a new alternative route, because if someone finds some medicine which is an alternative to the insulin, it will be a very a new era of the medicine to treat the diabetic patient around the world. So also he was claiming that it is a novel cisplatin protein, but this protein correspond to some other proteins called as PBF, which other scientists claim was a cardiovascular marker, which is found in another which is found as a biomarker as in the cardiovascular diseases. Talking about the model system, he did all the experiments in the two models, male and female, but he published the data of only the male mice, not the female mice. And the reasons were not given. I tried to find it on internet, but as the dean, they said they don't wanted to do the report public, so they hide the data. So actually, hiding of the data is also, I think, considered to be a fabrication of data, because you're not providing the whole results. So you cannot conclusively say, okay, this things works and this not. And they didn't tell anything about the clinical trials, and they just claimed that it can be a new target for the discovery. So maybe the more clinical trials could have provided more evidence, but as if this person, he's involved already in the other kind of problems with the reproducibility, so he didn't provide correct data. And I think the good role was played by the university in this case, because they cross-check it, because it's very rare that the universities play any. Generally, when you send up article for the public publication, the reviewer, it's very important to ask important questions, to get to know what exactly people are hiding and what they are presenting in the data. So the university here, I think it's play a very crucial role here, because they cross-check with the experiments and they set up a committee and they did the whole thing, and they asked them to retract the paper. So yeah, so I think this is my whole sum up of the article.
Great, thank you. So again, not clear at all that there is fraudulent science here, but there is definitely not good enough science. Cherry picking data that, you know, fits your interpretation is a big no-no. I mean, you have to consider the whole data set and basically discuss the whole data set. If you cherry pick and you don't show everything, then it's almost falsification. It's not really at that same level, but very bad scientific conduct. The paper is open for discussion.
I just want to make sure if I understood correctly. Did they prove there was misconduct? Like, did they prove that the data wasn't real, that it was fabricated?
The dean said that they clearly mentioned that the data was not fabricated. They said, we didn't claim that it was misconduct, but they said that they hide the data, like it was not a complete report to make the things conclusive. Maybe this thing doesn't work out in the female mice. So like just they pick up the positive data and publish it, which is also kind of misconduct, but not exactly misconduct. So they just publish half the data. The female data, they didn't show anything about it.
So again, it goes back to control. Good scientific conduct always have the experiment and the control has to be really, really well thought of and relevant to what you're trying to show. So hiding some of the data is kind of not having a control in a way. Other comments?
I have a question. Sorry, maybe I misunderstood you, but why not publishing results on female mice is hiding results? Because it is common to use only male mice in order to prevent too much variance in the result. Maybe it's an old-fashioned way, but it's something that I believed was common.
Oh, so I will answer to you in a technical way.
Okay, but maybe before you answer, there is also a relevant comment from Hanan, and he says, if they were stating in the text itself that the results are only for the males and not, be general, that might be okay as well, because they disclose that fact. So
like some of the committee members, I don't remember the person name from MIT. He said,
sorry, it's not Hanan, it's Hannah. Sorry. Yeah.
So one of the person and editor-in-chief, that he said that it's okay to not present the data, but actually in the report, the full article with this news was published, they said that when they produce the male mice, the difference of the new protein, this visfatin is very nice. But in the female, the level of visfatin was not very differences in the normal and the wild, the deficient and the wild type. So basically they are hiding because they wanted to prove their theory.
Okay, because so you say that they did check it, they just didn't report it.
Right. They did not disclose that.
So this is what I was
thinking. They checked it.
Okay,
they checked it, but they don't want to publish it just to make their theory correct, prove right.
Thank you.
Okay. More comments?
I have a technical question.
Go ahead.
How do they know that the visfatin was interacting with the insulin receptor? How did they measure it?
They measure the binding affinity of the visfatin and the insulin to the receptors, and they mentioned it was 4.4 in the case of visfatin, and its insulin it was 6.1. So it was nearly very close to the they calculate their dissociation constant by using the binding affinities assays.
But their purified protein preparation was not stable and was not.
They said only 4 of the batches work. They produced 4 successful batches, and that said that they are willing to set for the validation. But they said, but they themselves contrast, like they said that not all the preparation will work. So it was like fishy, because if the protein is good and stable, it should work in all the condition and other, like it should reproduce in other systems and everything, I think.
Leon, you had another comment?
Yeah, this is just data. How do they know the mechanism, the biochemical mechanism?
No, they did experimental assays of the binding assays of these two proteins.
But did they see activation of signaling once they administered the purified protein? No.
They see that the lower part of the receptor got phosphorylation, because when the insulin or the vispatin bind to the receptors, the lower part gets phosphorylation, and they checked it with Western blot, and they said that there was presence of the phosphate. So it was activated, the channel was activated, and then they checked the activity with the binding assays.
In principle, that part was correct. Okay, any other comments? All right. If no more comments, I ask John, David, and Radhika, please send me your PowerPoints and your PDFs. We are going to meet again next week, and whoever is speaking next week, please line up.
Shimon, I have a question. Do we have any deadline for sending 2-page report? Like, or we
no, it has to happen before the end of the semester.
Okay, thank you.
Next week, I'm still here in Los Angeles, but in 2 weeks' time, I'm actually in Israel, and I'll be in Tel Aviv, but I don't think we are going to change the time, so it's still going to be a Zoom. I would love to meet with you in a classroom, but I don't think we can do it at 7 p.m. So anyways, we'll see you next week, and looking forward. So far, I find those presentations terrific and interesting, and I learned from each one of you. So thank you. Good evening, everybody.
Thank you.